All right. We appreciate every one of you who made it available tonight, and I want to say thank you. I also want to appreciate those of you who join us in the webinar session on Saturday. It was really a mind-blowing one, and yeah, it was a mind-blowing one, and I want to appreciate you for that time. We really know that it is not easy, coming up most every day by 8 p.m. 2 hours to learn. It is really not easy, but it's a sacrifice that we are paying. So I want to appreciate you. I really need to appreciate you, because if you are not there, then the webinar wouldn't have been a success. Imagine that we just come up and then there were nobody present. So it would have been somehow. So I really appreciate you for coming out to learn. And I really know that the knowledge you are gaining will be applied. Yes, one day it will. You will see the proof of the knowledge you are gaining. So quickly tonight we are going to look at what we have. I'll quickly share my screen. So, are you seeing my screen?
Yes, sir.
All right. Okay, thank you. So we have succeeded. We have succeeded in phytoinformatics. We did that, and we have been exposed to a lot of things in phytoinformatics. So we shall look at another aspect of the training, which is chemoinformatics. So I want to ask, from your assignments, of course, the assignment has exposed you to a lot of things and you have learned a lot. So do you have a question? Do you have a challenge? Do you think you can share with us? Is there any question, anything you want to ask in regards to phytoinformatics? Okay. So the absent means
sorry, the absent means he shows one database. Yeah, we can also generate
Hello,
can you hear me?
Please, if your mic is on, off mute so I can hear you. Okay. My voice is Can you hear me now? I can hear, but is echoing.
It's from your settings. So.
Okay.
So there is one of the database.
I can hear you clear now. So yeah,
so whereby we can also I want you to remind me that database where we can also generate the admit property.
Okay, impact.
Okay, impact. Okay.
Yeah.
Yeah, good evening, House. Mine is a little clarification on the assignment.
Okay.
Yeah.
Which of the assignment?
Well, the one assignment for the phytoinformatics.
Okay.
Yeah. I observed that for my plan that I'm working on, especially in the literature review, some manuscripts using the same type of what's it called, extraction or characterization method, the results are very varied. I mean, I'm not a chemist, but again, does this throw some doubt on the authenticity of the results, or it's a possibility? Sometimes in some there are overlaps, but in others it's completely new. That is not even found in any other database. So, and again, my worry is I'm not a chemist, but using the same result, I would have expected some commonalities, across the manuscript, but that is absent in some cases.
Okay, if I get it clear, in the literature search, right, in those manuscripts, they used the same, let's say, the same method of extraction.
Exactly.
Right. So let's say ethanolic extract. Maybe they use ethanol as a solvent for extraction.
Exactly.
And then they did characterization
by probably LCMS or GCMS.
Fine. And you notice that their result is totally different.
Exactly.
Good. So thank you for that observation. Okay. Now we will not expect the result to be the same, but we expect to have some similarities. Yes, we expect to have some similarities. I don't know. I would like to come from the angle from your own discipline. What is your discipline, sir?
I'm a pharmacologist.
Okay, pharmacologist. Okay. All right. Now, those who are in forestry, in biotechnology, they will tell us about biodiversity. So if I collect, maybe we are working on, let's say, you are working on Agerantum conazole as a plant, or you are working on moringa. Then, if I collect moringa leaf, currently I'm in Imo State. So if I collect moringa leaf in Imo State and conduct characterization with ethanolic extracts, and then while you are in Oshobo, in Osun State, and you also perform the same moringa leaf ethanol extracts and did the characterization, we are not expected to have the same results. We may have similar because of biodiversity. The constituents of that same moringa here and the one in Oshobo will not be the same. There will be a little variation. So if you observe differences, they are right. We don't expect them to be the same, but there will be similar compounds. And also, it now depends on the plant part, but for the question, it means it's the same part, the same leaf or stem. So another reason why you have a different result is due to the GCMS, the kind of machine they use, the setting. Let me share with you an experience I had 2022. I sent a work. I won't mention the lab, but it's common, it's popular in the West. I sent that result. It's actually on Medicago sativa, alfalfa leaf. So the result of GCMS we got was purely on halogenated compounds. So I don't know how they programmed it, that the GCMS result only provided halogenated compounds that has halogens from that our extract. So we doubted the result. We now sent it out. When we sent it out, the result now, that same thing to another place, the result we got was purely phytochemicals. So it now depends again on the programming of the machine. Yeah, on the programming of the machine and the solvent, because the people who will run this GCMS, they will also use a solvent. So they also use a solvent for it. So it also depends on the solvent they are using. So these are the few reasons that will lead to that difference in GCMS result. So we don't expect to have the same thing. So
yeah, thank you for the clarification. One question that arises from that, well, yeah, as you said, yeah, there are some overlaps in some cases, Whereas in other cases, the variation is so big that you begin to doubt, if this can really be that. But with the explanation, fine. But again, another thing that comes out from there, is the issue that people have always raised with natural products in terms of standardization. So if I'm developing, let's say, a compound from a leaf I've gotten from, let's say, my own area of abode, and I discovered a very wonderful compound that has a very promising, a very promising value, therapeutic value. By the time I go out with this, and other plants in other areas are not having what mine has, or even will I even be able to reproduce that same plant? You understand, in sufficient quantity, that will make sense for drug value. So these are issues that people have been raising about.
I have a solution to that issue.
Okay.
I have a solution.
All right.
Yeah. In my organization, we are currently working on a drug for ulcer. Now, if you want to go into this formulation of herbal products, then you must stop collecting samples randomly because of what we just explained now, and that is the issue too you have raised. That's the challenge. Like for instance, you are using bitter leaf now to produce a drug, a herbal product, and the bitter leaf in your area, you have exhausted it. You now made contact, and they told you that there is bitter leaf in Lagos. You collected bitter leaf from Lagos. They told you again there is a bitter leaf farm, very very big farm in Abia State. You go collect. You are making a very big mistake, and that is the challenge you just raised now. You are making a very big mistake because your product will not be standard. So what we do is that you must have a farm. Have a farm where you cultivate these crops, your plants. Every industry that want to do well in herbal formulation must have a farm. Nigeria Agency for Natural Medicine, they have a farm. You must have a farm, a land where you are certain that, because if I have a farmland, if I control the farmland properly, keeping all parameters constant, if I control the farmland, then I keep planting this the plant of interest. I keep planting it at that same farmland. It will reduce the issue of variety in chemical constituents. It will reduce that challenge. And again, in fact, it's not even the difference in the chemical constituent is not even the issue. Another big issue is the toxicity. Sorry, but it's good. This issue is coming up because that is what we're discussing. We are interested not only in the therapeutic activity, but in the safety. Hence, from my experience, herbal toxicity arises from three factors. Let me just share it with you. Herbal toxicity arises from three factors. One, toxicity as a result of the toxic compounds in the plants due to differences in their geographical location where you are collecting these plants. They will have different toxic compounds in them. That is inherent. This one, the toxicity is due to the chemicals in the plant, is inherent. Number two, that land, the land where that plant is being cultivated, we also provide toxicity. We know about plant bioaccumulation. That plant can bioaccumulate heavy metals from the soil. So if you keep collecting plants from different locations, you don't know the anthropogenic activities carried out on that environment. So if you collect from Abia, it is possible that you are not only collecting the plant, but you are collecting plant that has been contaminated with heavy metals and other environmental pollutants that can be easily absorbed by the plant. You collected from Lagos, the same thing. So you are formulating a product given to somebody, and the person is accumulating all the heavy metals or other environmental pollutants being accumulated in that plant. So these are possible sources of toxicity, and that is why we are afraid of herbal products. But if you can have a farm and you control your farmland, you run your analysis, all kind of analysis on the land, you treat the land very good. Again, toxicity can arise from the nature of the fertilizer they are using. These fertilizers can deposit, they can be that they can stay in the soil for a long period of time, and some of them are toxic, and the plant will also absorb them. So we are not just interested in what the plant can do, but also the safety of using that plant. So the solution to all this problem is have a farmland. We have a farmland. We have a farmland, but we don't focus in anyhow plant to that is personal to us. There are some kinds of plants. Imagine you're working on tree, mango tree. Are you going to plant mango tree? How long will it take for a mango tree to grow? If you have to focus on the stem back. So if you have to have a farm, you have to select the kinds of plant that you want to work on. So the solution to this is have a farm. Thank you very much, sir. Hello, sir. Okay.
Yes, thank you very much. Thank you.
Yes. Even I will encourage all these schools who are bringing this, the department presented here, mostly the ones on the pathology and the pathology. When you go back, have a farmland in your school. Yes, we have said, herbal products is the way out. You see in Nigeria, and it's very easy to be approved. NAFDAC can easily approve you. So we are looking forward to have numerous herbal products very soon. All right. Is there any other question and contribution from our previous class? I love this outcome. It's very good. Okay.
Hello, sir.
So yes.
Sir, I wanted to ask this in Dr. Duke's, when in relation to the assignments, because most, like the plant I'm working on, when I clicked on it, like to find the metabolites and all of that, I was having, because I don't know, in the previous classes I attended, we explained the Dr. Dukes and other distinct, but here I could find some bioactive compound that they would say maybe the activity count is 0, the low parts per million, there's nothing, high parts per million, and then why some will have and the rest. Do we download those metabolites that are under the plants? Like, do we just download it, or we have to look at those categories that are listed in the columns?
Okay. Just the download all the metabolites, even though that some of them has activity 0, those stuff 0. Yes, download them, because whatever result they have there is due to past records. Yes, is due to past record, and they did not specify which receptor. They did not specify which receptor. So that a study was done with, let's say, with aspirin on diabetes, and there was no activity. We will not just include that aspirin does not have any activity. There are thousands of diseases, thousands, millions that have not been tested. So I will encourage you, our interest is get the metabolite, all of them.
All right, sir. So sorry,
Yeah,
okay, this is like a continuation of the question. For the because some of the metabolites in the Dr. Dukes, because I didn't there was no other alternative aside the pop chem. That was, I don't know if you had given another alternative that where we could get these compounds, because some of them, especially in that Dr. Dukes, like I found a lot of metabolites for my plants in that Dr. Dukes
that are
more
not in
than
pop chem.
other. Yes.
Yeah. That is the challenge. You can, okay, I think we are working on today. Today we now expose you to other database. Yeah, today we expose you to other database. But when it comes to in silico studies, PubChem is well, in fact, the way we rate impact as number one in phytoinformatics, that is the same way we rate PubChem in Chemoinformatics. But there are other database too, which we're going to look at today.
All right.
But then, if you could not find any compound in PubChem, it's already a red flag that you may not get it in any database. So it's already a red flag because these databases they are related. They are related.
Okay.
Yes, they are they are connected. So, but we have KMBL. I will expose you to those ones today. So if you don't find them there, maybe you now search in other database and you could not find them. The only option now is to design that compound yourself. We did that in our free training last 2 weeks. Yes, last 2 weeks we did a training on macrophore modeling, where we learned how to use sketch to design molecules, because we have noticed that some of these molecules are not found in this database, and this the database are manually deposited. So maybe they have not deposited those ones that you saw. So you have to now draw them. You have to look for their structure. Maybe copy the name online and say structure of so compound. Then you get the structure of the compound. Then you use your chemistry software, your structural software, to now draw that structure and then convert it to 3D, optimize it, minimize it, keep it in mole in SDF, the same format in which you download from PubChem too. You can keep it there. And we are having a training specially on this by next week, 28th. It's starting 28th. It's about 2-3 days training. The flyer, you must have seen the flyer, is a beginner's course. Yes. So I will encourage most of us who wish, in fact, it's for everybody here, because there are cases where you may not find your plant. You may not find your plant in this database. You have to design it. So that beginner's course is purely on drawing of chemical structures, minimizing them, optimizing them, converting them to 3D, generating other identities. So it's just for 5,000 instead of 20,000. So you can sacrifice that and also join. Is last week of January, which is next week. It's starting next week, Wednesday. So you can enroll in that training. I will share the flyer with us tonight. Thank you for that observation. So now that it becomes tedious now. That is where the word is. It becomes tedious. Okay. All right.
Thank you, sir.
So at this point, we are going to look at what we have for today, which is chemoinformatics. Phytoinformatics has been a wonderful.
So sorry, sir. I've been raising my hand for a while.
Oh, sorry, I'm on my screen. So if I'm not on that page now, that is why I could not see you. All right, go ahead, sir. Sorry.
I'm sorry. When we were talking about the diversity in plants due to their different geographical locations, I'm wondering if the diversity will play in when we are doing purely in silico research. If you are not doing wet labs, would the diversity still be significant in the wet lab? And then concerning the assignment, you said we search 50 literatures. Are we downloading the papers, or are just referencing the paper that discovered the bioactive compound?
If you let me come from the last. If you don't download them, if you don't download the paper, then you may not, if you find it difficult to download the bioactive compounds in those paper, because our target is not on the paper, but on the bioactive compounds discovered in those papers. So that is why you need to download the 50 papers first. Then start opening them one by one, and then look at the results and download their compounds, those compounds in those papers. Download the compounds. So that's why you need to download the paper first. All right. Then this question is very interesting. Your first question is very interesting. The diversity, relating it to wet lab and in silico. That is a very critical point. You know, I've not really looked into it in silico. But let me say this. I think I will be realistic now, due to we are not just learning this for let me learn to know how to do in silico, but our aim is to develop a product. Fine. So in silico, we are just getting the bioactive compounds. We are not even taking records of where those work, because we are just downloading it. So after downloading those compounds, we don't even know the papers that reported those compounds, because you're not building a database. You're not building a database. But if you want to build a database, that means to every compound you download, you
We can't hear you again, sir. I hope I'm not the only one.
I want to if it is even my network, because the thing has been fluctuating.
Me
I
too. I'm
don't
not
know.
hearing him again.
I want to be sure if it's for the
Is
so
everybody?
I think because I'm even
Is everybody?
outside my house now looking for network. So which means it is from
Is
it is from
everybody
his
off?
end.
I can't. Is the mic went off? I think it's a problem from his own side.
Okay. Actually, I have problem with this Google Meet. The Zoom is better.
I think the problem is from his own end.
Okay, what is
Hello?
happening?
I cannot hear anything too.
Up till now? Oh, okay, that means there's a problem with his network.
I think it's his own
Is he
network.
back?
No.
That would be network from his own side, though. I think when he's back, we should try to suspend our questions till after this the training for tonight. So this is almost tonight. You suspend the remaining questions.
Okay. Hello, can you hear me back?
You are welcome back, sir.
Yeah,
Ah,
so it's now.
oh,
You're
so
welcome.
sorry. Yeah. So sorry. I I was talking. I never knew that my network disconnected me.
Okay, I was wondering.
So yeah, the question our friend raised is a very good one, because since we are not taking note of, since we don't know the location of where these compounds we are gotten, and we are just retrieving them, we are doing historical studies, there's a probability that if at the end we discover that benzoic acid from all the database now had a better results, very stable after MD and everything, then we decided to validate it by doing wet lab. We then collected that plant from our location. Don't you think that it is possible that at the end, the plant we collected in our location may not even have the benzoic acid because of the diversity? Don't you think so? That's the question he was asking. It is possible. So what is now the solution? The solution is, if you want to do a product development study, what we are doing here is training, teaching you how you can screen libraries of compounds. But if you want to do a product, your target is, I want to have a product. That means that you are not going to be using database. You are not going to be using literatures. I don't know if you can hear me.
Yes.
So if your target is, I want to have a product, it means, one, have a farm, cultivate that plant. What the initial process you will just do is to use this database, literatures, to have a clue, to identify a compound that, from what I have done, based on literatures, based on the compounds I got in literatures, and based on the compounds I got from a database, compiled together, I did these historical studies, and I discovered that about 10 compounds here are good. So that means it's a green light. I can proceed to cultivate this plant in my farmland. So now have a farmland. Cultivate the plant in your farmland. Then decide what what kind of formulation do I want to do? If you want to base your work on just the herbal, the powder, or you want to base your work based on the extracts, maybe an extract. So if you are using aqueous, you use aqueous. Or you can now, this one now, you are the one conducting the study yourself. Let me tell you, if you are developing a product, you will not rely on other people's results. All the results you must have must be generated by you. The characterization results you are using must be the one you conducted by yourself. Because you cannot come to me and tell me that I have gotten a drug. These are people who have reported it. No, we need to have your report. So it means that you're going to perform your own characterization now. Work with your own results. Because it can be easily validated. When you now go back to that same farm, that's your farm, and collect the leaf to validate the in silico studies. Because if you don't do so, if you are relying on database and other people's work, in product development, you're your own. Part of the 3 months, we are organizing a 3 months professional training on herbal product development. And these are part of the things we prepare to teach in that 3 months training on herbal product development, combining in silico and wet lab. So you must rely on your own results, because using database and literature, at the end, if you want to validate it, the location where you're collecting these plants may not contain that same bioactive compound you have discovered from in silico. But when you have your farm, grow your plants, do your own characterization, work with your results, then it is reproducible. Thank you. So the way we are asking, it means that we are interested in having a herbal product.
Of course.
Omo, it's a good one. Oh. Thank you very much for that observation, for that question. Okay. So some persons will say, I thought this is a hands-on training. We are talking so much. Let us start pressing laptop. But I have this slogan that hands-on is not difficult. The background knowledge is very important. I was trained in a polytechnic in my first degree. We don't go to the lab for practical when we don't have good theory knowledge. In fact, our lecturers then will tell you go back for your theory class before come to the lab. So the laboratory work is a very simple thing, but the theory knowledge guiding you is very important. The knowledge guiding you, backing you up. Everybody can come and do the practical, following the procedure. Miss this, pour this, measure this, very simple. But what are you missing? What are you measuring? What are you pouring? That is the foundational knowledge. All right. So today we shall be looking at the chemoinformatics. Permit me to continue, right?
Go ahead, sir.
Because we have taken is good. We have taken almost first 1 hour, but is still the reason for the class. So we shall I will be summarizing this aspects so quick. So we shall look at what is chemoinformatics. You know, just like the name is chemoinformatics is that aspect of science that combines the chemistry knowledge, computer science, mathematics, and information technology to solve chemical problems. So here we combine the knowledge of computer and IT, computer, chemistry, and mathematics to solve chemical problem. Now, chemical problem related to chemical substance. So in summary, it provides information, any kind of information you want to get regarding a chemical substance, regarding a chemical compound. You can get those informations through chemoinformatics. It's just like what we have in bioinformatics, why that one is for biological data, why chemoinformatics is for chemical data. You know, chemoinformatics has not been pronounced so much as bioinformatics because it has been it has been on the low for long, but it helps us to study, manipulate, retrieve, do all of what you want to do with your chemical data. It plays a critical role in drug discovery. Yeah, it has a role to play in drug discovery, material science, environmental chemistry, and agrochemistry, among others. I'm just going to be fast. Now, the overview, everything about it, what is involved in chemoinformatics.
I'm not seeing your screen.
It's
Is
from
anyone
your
see my
side.
screen?
We can see it
All right.
clearly.
All right. So now this natural products you
Oh, we
are.
can't see the slide. Oh, we can't see the slide.
Is anyone see
I
my
can see
slide?
it now. I can
We
see
can
it.
see now.
And
We can see.
we can see your
Now.
screen.
That is on.
We can see very well.
All right.
Yeah.
Oh, thank you. So now this natural product you are mining from this plant, the natural product you are mining from the plant, they are chemical substance. And then for you to exploit
Sorry,
them,
can't see us.
that is work from your end. Is well. Don't worry. If I be your president,
It's appearing.
yes, if I be your president, this is the first problem I will solve in Nigeria.
That's how everybody have been saying it. And
No, I'm not everybody now. At least you have known me for a while now.
Thank you very much.
Until you climb that position, that's when you will know.
There's no problem. So since you are dealing with natural products, go help us. Since you are dealing with natural products, these natural products are chemical substance. So you need the knowledge of chemoinformatics to be able to know how to handle, manipulate, retrieve any kind of information you want to get about those natural products. What we did in phytoinformatics was just to get the phytoconstituent in our plant. Now, what kind of information do you need from these phytoconstituents? What information do you want to get from there is now from chemoinformatics. So chemoinformatics will provide you those necessary information that you need from those natural products. So in overview, it involves data storage. Now, we study chemoinformatics with the use of databases. So we store data. So it involves data storage. What data are you storing? You are storing those bioactive compounds you have modeled. Like I just explained about when you can't find your compound in PubChem or any other chemoinformatics the database, you now have to use structural softwares to draw the compound and store them. So that's what you are doing is chemoinformatics. It involves data. Then it involves indexing. When you download the compound from PubChem, it comes with the CID. With the CID, all of you have done that. So it comes with the CID. So those data you are storing must be indexed. Like the database we are creating in my team, the database we are creating for Nigeria, it has index. So when you download it, it will download with the index. Now, in the training next week, I will show us how to actually save a compound such that it will save with the name. Let me ask this question. Has any of you done docking? Like you have done docking with a compound you do by yourself? Has anybody performed docking with a compound you do by yourself?
Yes, I have.
Yes.
Now, when you perform the dotting, was it showing the name of the compound you saved?
Yes, I've done it, but not working.
Okay, yours is not working. Okay, but who has done dotting and it worked with the compound you do by yourself? Dotting.
No, it's showing the
Okay,
chemical formula. Showing the
Mine
chemical
worked.
name.
was it showing the chemical name of the structure you do? Okay, now
Yes, it's showing the chemical name.
Now, most times, okay, most times, if when you draw a compound, it's not just enough that you draw a compound and save it with the name. If you open that compound in probably if you open it in any of these visualization softwares like Pymo, Discovery Studio, it will be showing fragments. Sometimes to show molecule. Now, that means that you have not indexed that compound. So we attend to those challenges because I've also noticed that earlier. So we attend to indexing. How do they make this such that if you are downloading this compound to show with their index, not just because they saved it with that number, no, it was actually indexed. You can use Notepad to do that. It's very common. It's very simple. So it involves indexing. You index the compounds you are creating. The essence of teaching you chemoinformatics is not just for you to play around with this database. Have a vision of creating a chemoinformatics database. Assuming this class is expanded, like a 3 months, 6 months class. Part of the job we have done, the assignment is to group us in 2s or 3s. And each group must create a mini database, at least with Excel, or just a mini database that can be published. Because when I teach chemoinformatics, my goal is to enable you learn how to create a chemical database for yourself. Not the one you store in your folder, but at least you can use Access or Excel just to create a little one for yourself. So it involves indexing, but because we are just centered on macroglossy, so that is why I won't go into that extent. Then it involves search. There must be that access to search for those compounds you have stored. So in this chemoinformatics database, it gives you the ability to search any kind of information you want to search. Now they are of two types. Some of them work by deposited compound, while the other work by algorithm based on machine learning. Like the PubChem works mostly by deposited compounds. So any compound that is not deposited in PubChem, you can't access those compounds. But if you use AdmetLab, SysAdmet, they don't work by deposition. They work by algorithm. Once you input the SMILE code, they use the SMILE code and generate the information that have been programmed in that system. So we have the ones that work by algorithm.
Your screen is very challenging.
Yeah. Then it gives you that privilege to retrieve. You can also retrieve what you have saved. So these are the overview of chemoinformatics. Then it gives you the ability
your screen?
You can't see my screen?
Honestly, I can't. So.
Can't see it.
It's from your side, though. Can see
Your screen is very
clearly.
All right. So it gives you the ability to also analyze chemical data.
My village people.
I just want to run so fast with it. Now, there are some areas in chemoinformatics. There are key areas we look at in chemoinformatics. We look at the one area is molecular representation. So you learn how to represent your molecules in 2D or 3D. You draw 3D structures and deposit them in this database. You can use tools like ChemDraw to draw them, minimize, optimize, and deposit. Then you can keep them in different file formats. You can use mole, SDF. Mostly is SDF. You can also use the mode file. Then you generate SMICE code for them. Then you can also generate descriptors. Now these descriptors are properties as a result of the chemical structures. Stuff like the molar weights, the log P for solubility, the hydrogen bond donor, hydrogen bond acceptors. These are called the chemical descriptors, and where we use them is mostly in ligand-based drug design. If you are doing 3D tracer, we apply these descriptors. So you also generate these descriptors and store in this database. They are all under the molecular representation. Then we have database management. These are another key area. It gives you access to manage your data by storing and managing large data. An example of those ones are the PubChem, the ChemBL, and Zinc. We have more than 100 chemoinformatics database. More than 100. Yeah, but it all depends on what you want to do, and the one that gives you what you want. So you can search as much as possible. Then it also enables us to do chemical structure searching. Like in PubChem, you can search for exact match, you can search for substructure or similarity search. Now, one thing that you can do is that most of us always use PubChem to download our compound, but that is not the only thing. You can use PubChem to search for analogs. Yeah, we'll look at it once I just round up my slide. You can use PubChem to search for your analogs. PubChem to search for compounds that have similar structures, which is very important if you are dealing on ligand-based drug design. It comes in there. If you're working on ligand-based drug design, then you must use compounds that have similar structure with your reference drug. Another area is the modeling aspects, where you have to predict molecular geometry and energy using force fields. So I think we'll be doing a work on this by tomorrow. There is a 2 days free training starting tomorrow, just Wednesday and Thursday, on molecular modeling. So that is where we can discuss more on this. Then another area is your docking, homology modeling, and MD. One major area is you can use it for QSA and QSPR. I've already explained that. QSR means quantitative structure activity relationship, while QSPR means quantitative structure relationship, structure property relationship. Now, what that means is that compounds with similar structure, we have similar activity. So compounds that look like paracetamol, we exhibit similar activity with paracetamol. And also compounds that look like paracetamol, we exhibit similar property with paracetamol because they have similarity in their structures. So we can use our chemoinformatics to retrieve those compounds that have similarity in their structures. They're often used in drug design, and even too, we can apply QSAR in toxicity, because compounds that are similar to, we possess similar toxicity. So that is the idea of QSAR. And we're having a training on QSAR by February. In the first week of February, we have 2 days free training on QSAR. Another area in chemoinformatics is virtual screening. You can use them for virtual screening. Initially, when I started docking about 8 years ago, I was just using web servers. I was using a CBDoc. Yeah, I was using CBDoc for docking. And I actually did a work for somebody for a master's degree work using CBDoc, and it was approved. Till now, I'm wondering how come. So we can use them for you can use these chemoinformatics tools for virtual screening. You can screen large compounds to identify potential lead. And this is what we do in natural product mining, where we can screen large compounds to identify potential leads. So I've already explained about database. One of the applications of chemoinformatics is drug discovery. We can use it to identify our hits and leads. So when you have a library of compounds, with the knowledge of chemoinformatics, you can identify your hit compound. Then you can, from the hit, get your lead. So let's take, for instance, that my hit, one of my hits was benzaldehyde, one of my hits. And you know, from your hit, you now optimize your hit. So I can use my chemoinformatics tools to get analogs of my hit. So from there, I'm doing optimization, because in optimization is of two ways. It is either you are generating derivatives of that your hit, or you are reducing the structures. Maybe the structure is too large, and you just have to identify the parts that you are involved in that interaction, then take off the other parts that we are not involved in the interaction. So what you are doing is that you are trying to reduce the size, maintaining the active site for interaction. So this is what you do in the lead optimization. Then you apply it in SAR studies, which is what I explained about structure activity relationship. I just want to round up so we quickly look at some of those databases. Then we use them for ADMET prediction. Now we will discuss more of this in macrokinetics. There's a debate on how well can we trust this computer-generated result, mostly the ADMET properties. How well can we trust them? Because they were generated by algorithms. So how well can we trust them? Are they valid? Can we still reproduce them in the lab if we are doing wet lab? So we'll discuss on that challenge when we get to macrokinetics. So we can use them to generate toxicity. There was a work I did. That work was presented in a PPSN, in the Public Health and Parasitology Conference. So it was actually on insecticide, because we noticed that some of these insecticide we have around us, they are toxic. So I did that work with garlic, and I discovered ajoin. Ajoin was very good. I used 24 insect-related protein, 24 from different mechanisms. So ajoin was very good, but from the toxicity, from the eco-friendly work we did in silico, we saw that it has skin irritation ability and skin sensitization ability. So you can use chemoinformatics to generate those high-profile toxic models, to toxic prediction. I have some softwares that can give you up to 30 toxicity models, about 30 when combined, which is very good, because that is where we are after. So we expose it into that when we are doing our toxicity screening. When we get to that model, so we'll do that. Then these are some of the data sources for chemoinformatics. We have PubChem. So we can start with PubChem now tonight. We shall introduce you to PubChem. We are almost familiar with the PubChem, so I will just show us other things we can do with the PubChem. We have ChemBL. That is second to PubChem is ChemBL. We have Zinc database. Like I said, there are over 100 database for chemical compounds. I use them depending on what I want. So, and please don't ask me to share them with you. You can go online and just ask your Google to list all the, maybe list 50 chemoinformatics databases. It will provide them for you. Then you begin to study them one by one. you know their merit and demerit to know what they do and what they cannot do. That was how I discovered them. Then Drug Bank, I've encouraged us, those of us that have our institutional email, to register with Drug Bank. You can use it to get information of drug, FDA approved drugs. Mostly if you are interested in drug repurposing, you can just lift 1 million of drug from there and repurpose them for another disease. We have Binding DB. These are the other databases. We have Swiss AdMe. We have TOSNet. We have AdMet Lab. And so there are too many. So these are the one we shall be exploring tonight. Then we have challenges in chemoinformatics. Our challenge are quality and data standardization. So most times these data we are collecting mostly from the depository database. Most of them lack that quality, so it's a problem. Then you have a challenge of handling large data sets, big data. Though from cloud computing now, cloud computing can handle that challenge of big data. Accurate 3D conformer generation is another challenge we have noticed in the chemoinformatics. The integration with genomics and system biology. Those who are very familiar in bioinformatics, we are now talking about multi-omics. That is where we are in bioinformatics. We are no more talking about genomics or proteomics separately. We are saying if you want to do a genomic study, then you must employ multi-omics because they are related. Then we don't have a database that can cover both the chemoinformatics aspects and bioinformatics. So the integration with multi-omics is a big challenge. Then interpretation of the black box model for machine learning. Most time these results we are generated by algorithms. What happens in the process becomes a challenge for us to interpret. What we only see is the result. But the process, the black box model that was created, how valid it is. Can we interpret the model? We don't even know the model. What we know is put in your smart code and you get your results. We'll leave that for those who made the system. So we have a future. The future is AI and deep learning. This year we have training on machine learning, but next year we are putting up a training on deep learning. Deep learning is higher than machine learning because it has multi-layers. Yeah. Most machine learning just uses 2 or 3 layers, but deep learning makes use of about at least 5, 6 and more layers in the black box reasoning. So it helps for better prediction. So by next year, there will be an intensive training on deep learning. So if we can incorporate AI into our databases, then it will be better. And that is what people are doing currently now. And the use of cloud computing is the future, which we have been discussing. The expansion into green chemistry. We have started work on green chemistry. And that is our future. Even in medicine, yeah, green chemistry is still the future. And we did a training December last year on green chemistry. Very wonderful training. And another one is coming up by February. The advert has been on green chemistry. It is okay. People have not been actually. It is rare to see a work on green chemistry that has in silico. So we look at that limitation and we decided to train people on green chemistry using computational tools. So it is still novel. That area is novel, is novel, and that is the future. We have a 1 month training for that, starting by March. Sorry, by February next month, 10th of February. So that is where we are going to. So today, let's start with the first. Let's start with the first database, PubChem. So let's look at PubChem and what we can do with PubChem. Okay. Yeah, are we together?
Yes, sir.
Yes, sir.
All right. So let's look at the first database, PubChem. One we wonder that we started and we have been talking about database. Why? Why is this so? Uh, I also wondered so because we have been centering our discussions on databases. The reason is that the success of in silico work depends on data. All in silico studies depends on data. And that is why you must be well familiar with these databases because they are the source of your information. So you must be well conversant with them. Network should be fast here because I don't really know. Oh, this is Nigeria.
Very frustrating.
What is happening now? As I'm telling you, you type in something, you will not even see what you're doing. And what am I going to do now if this does not respond? So I can't really tell what is happening, but let me just change my. I'm just wondering why the network happens to be so bad tonight.
It's the weather. I think the weather.
Yes, weather is another thing. I just changed my network source. Oh, what are we going to do now?
Nigeria.
All right, okay. Can you see my screen? All right.
We're fine.
All right. I think there's a solution for us now. If you can see my screen, let me know. Can you see my screen? All right.
Yeah, I can see us.
So you see why I told you that I'll be your president, and we'll be using 10G. 10G.
It's well.
So what we'll do now in PopGame is that I'll quickly use what we have now in the next 30 minutes. So let's do that together now. The first thing in PopGame is your exact match search. You search for compound. Now PopGame is not only used to search for compound, you can also use PopGame to search for plants and their bioactive compounds. So I think I will do that first, using PopGame to search for bioactive compounds of plants, like as we did the impact. You can use PopGame to do that. There are some plants that has been deposited also in PopGame, because when they deposit a compound, they have to provide the source of that compound. So if the compound has a plant-based source, so that plant must be identified and included in the database. So from that algorithm now, you can also search for plants and their bioactive compounds. Okay, so let's say we search for moringa. So you can type a plant. You can type a plant. So when you type the plant, you come to taxonomy. When you type moringa, now whatever you type here, there is compound, there is gene, and there is taxonomy. So you can find Moringa oleifera. You can go to taxonomy and look for Moringa oleifera and click on it. So I'm coming from the area of Chemoinformatics using PopGame now. Then I will complement it with Chemoinformatics. Now you see that it has only 2 records. Under taxonomy, you can find just 2 records. 2 records. You can find just 2 records for that. Now, those 2 records are these 2, Moringa oleifera. You can then click on any one of your choice. When you click, it will provide the bioactive compounds present in that Moringa oleifera. You scroll down to data. You can scroll down to data. Okay. So in the metabolite section, so you can see some of the compound found in moringa from PopGame. You can, so here has just 8 compounds. No, this is page 1 of 8. Okay, so it has 38 compounds here. So there are 38 compounds in PopGame from moringa. You can add this also to whatever you have gotten from your database search. Now, but the beauty is I can download all once. You can download all the compounds once. Once, since you are using PubChem, you don't need to do individual downloads. You can download these 38 compounds once. For you to achieve that, you come to download. Are we together?
Yes, yes.
All right. So can you find download here? This is download under the metabolite. You come to download. You click on the download. Then there's supposed to be an option for the 5 formats you want to choose. There's supposed to be an option for the 5 formats you want to download. There's supposed to be an option.
Dr. Henry, there's actually an option for you. Go to the download, click on the download. You can see it, right?
Yeah, but if you choose,
there's a general one here on the right-hand corner. You download there.
Okay.
Yes. So yes, options of what you want to download there.
Okay.
So maybe you may choose comma separated values or something.
Okay, I was actually working on the SDF, so so you go back to manual, you go back to one after the other.
I've done mine now, and have options of CSV file.
Yes,
JSON file.
Yeah, I'm looking for the SDF.
Okay, SDF. All right. Okay.
So that because we need their data, then when you download the CSV to come in a commercial value, which is can open it in Excel. So I wanted that option for the SDF.
Okay.
So that would be easier for us. But since we couldn't actually look at it, so you go back manually. That is, if you are take them one by one. Now, do not don't get confused by this metabolite and the natural product. The metabolites is actually the ones present in the plant. Then when you scroll down, you will see natural products, and they're about 313. Now, these natural products, they are not just the compounds found in the moringa, but they are compounds found also in other plants. Not only in moringa. So if you want to concentrate the ones for your own plant, just leverage on the metabolites. Yes, just leverage on this metabolite and we can download them. So one of the use of PubChem is for phytoinformatics search, and we have just seen that you can take your time to look at other things. Now, another usefulness of PubChem, before I come to what we already know, is you can use it to search for. You can use it to search for your compounds that can target a disease. You can use it to retrieve compounds that can target a disease. So maybe you just want to get compounds. Chemoinformatics is not only limited to natural products. So I'm giving the overview, both the ones that does not have to do with micrognosy. So just what you can do it with PubChem. So you can search for compound related to disease. So you can just type breast cancer, or you can type breast cancer. Maybe you want to work on breast cancer. You can type the breast cancer. And once you type the breast cancer, you hit your search button. So you can search for compounds that are related to breast cancer. Now, all what you need to do is to go to bioassay. Go to this bioassay. Once you typed your disease and you hit enter, you come to bioassay. Now, in bioassay, there are a lot of literatures. These are literatures that has laboratory-based results on breast cancer. So you will see the number of compounds that have been tested against breast cancer. Like for this first one, we have tested substance 1. But you can sort it. You can sort by saying sort based on active substance count. Based on active substance count. So, because if you are looking for compounds that have been tested on a disease, then you should focus on the active substance, the active substance count. So that is where we usually focus on. It's still updating. It's updating. It may take some time.
Oh, sorry, sir. What did they now mean by active substance count decreasing and increasing?
Yeah, decreasing. Yeah, they will provide based on the number of substance there. The papers that has higher number of substance. You know, these are different lab work, wet lab work that we have done. So decreasing based on the count. Maybe a paper has 5 compounds, another one has 2 compounds, another has 3 compounds. So it will be sorted in decreasing order, which is from the highest coming to lowest. Then if you choose increasing, it will be sorted based on the lowest, then coming down to the highest. So it's actually taking time to update. It's taking time to update. That should be a Nigerian challenge. Okay, but if yours can do that, you can easily know the number of tested compounds. We are mostly interested in the active substance count. So then another thing you can do here, let me show you something. Another thing you can do in PubChem.
Sorry, I saw 9,008 in my own. Is it possible?
You know what?
Under active substance count, sort by 9,008.
Under active substance count.
Yes.
Yes.
I saw 81,000.
Good.
It's
So
1,860.
Yeah, you can actually retrieve those compounds. Let me come back. I would have been patient. You can actually retrieve those compounds. If that is your area of interest, you can actually retrieve those compounds. Like, let me just show you how to retrieve it now by picking anyone here. How to retrieve those compounds. So I will just all these ones are having just one. I need ones that has more than one. If I go back now, it will take me time. But let me. Okay, I will be patient with this and duplicate, then come back to it. I would have shown you how to retrieve those compounds. Because you are going to, if that is your interest, you can retrieve those compounds and then use them for your studies, for your work. Maybe you want to repurpose them, so you can also use them for repurposing. Well, let me just use a typical example to show you how to retrieve it. Let me pick this one here. Let's now assume that this is the report that has those thousands of active substance counts. Let me go to active substance counts. The difference between tested substance and active substances: compounds that were tested but they don't have they lack activity. But once it's active substance count, it means that those compounds have known activity. They have known activity, known biological activity. So if I click on this report that is giving me one substance count, if I just click on that your own now, click on the reports, click on the reports. Now, once it opens, I will show you where to download to retrieve those compounds. So, are you working with me now? Are we on the same page?
You said we should click water.
Click on the, for those that their PubChem was able to update. So click on those, the one that you have the highest number of compound count. Some of you recorded 8,000 something and all the rest. So just click on the literature. Look at what I did. You can click on like this, the write-up, anti-tumor activity against. You can click on it, or you click on the bioassay ID. The bioassay ID. So it will open. So once that opens, scroll down. Scroll down to table 4. Now, it is now in table 4, that you can find those active substance counts. Okay? Are you seeing that in your own?
Mine is table 5, 5 data table.
Okay, data table. Okay. Now, once you can see that data table, click on download. Click on download. If you click on download, now if you click on download, you can take the CSV. But there are two options here. We have data table all, data table active for active substance. So we are always interested in the active substance. So what I will advise you is this. Since you are unable to retrieve them in SDF once, because I've been looking for that, since you're able to retrieve it in SDF once, then what you will do is to take your CSV. The CSV will download with their smile code. It comes to their smile code. So choose the second option, which is active, the one for active. Don't choose all, because we're only interested in the active substances. So you can click on CSV and it will download as CSV, which can be opened in Excel. You can open it with your Excel. So you can open it to your Excel. Now the beauty is that you can now continue working with those compounds using the smile code. So from the smile code there, you can use them in your ADMET studies.
I think you can have the CID number is there also. Take it, go back, you can see it now.
Yes, it has the CID, but what I was looking for is SDF, where I can download all once in SDF.
But I thought it's one now. That one doesn't mean that it's more. I thought it's one.
No, I want to skip maybe if I want to do docking now, I will need the structure. So I want to skip that downloading one after the other, but I couldn't find it. So this is just one compound. Those of you that had about 8,000 compounds, all the 8,000 compounds will be saved here in CSV with their CID and their PubChem. With their PubChem CID and with their smile code. So the most important aspect is the smile code now. From the smile code, you can now do your ADMET studies. But if you need the structure, in structural data file as we have been doing. It means you are going to be downloading them one after the other.
Sorry, there are two numbers here. Which one is
Yes.
the CID?
This is, let me open. The CID is the second one. The first one is SID, the substance number. While CID is the compound number. The second one is your CID. The third one is your SMILE code. We have SID for substance, and we have a CID for compound. So the second one is your CID. The third one is your SMILE code. Now, another thing you can do with PubChem, I'm just showing you. Another thing that you can do with PubChem now is you can retrieve analogs. Analog. Let's say you did a work and you discovered that you have a compound, benzoic acid. Let me just use benzoic acid. And okay, let me say entrepreneur. I've been working on entrepreneur for long. Okay, let me say benzoic acid. And you discover that benzoic acid is good. It has a better result. So you want to check the analogs of benzoic acid. You don't want to draw them yourself. But I will always appreciate you to draw them yourself, because drawing them yourself, it will enable you to input the features you want. So it will enable you to design those analogs using the features you want them to have. Or if you are retrieving them from PubChem, that means you are just retrieving what has been modeled by other people. So let's say I'm working on benzoic acid now. You just type benzoic acid and search. Once you type benzoic acid and you search, now I will come to best match and all those stuffs. What you would do is you from this best compound, the best match, which is the first compound, you click on similar structure search. Normally, while you are downloading your compound, you just come to the CID and download. You click and go to download. But now we want to retrieve compounds that are similar with the benzoic acid. So you click on similar structure search under the best compound match. So once you click on similar structure search, it will bring up different kinds of similarity search. The first thing is the identity, which is 1. That is the best match. Then similarity search, substructure search, and superstructure search. So what is the difference between similarity search, substructure, and superstructure? Now, if you look at this similarity, it's telling us that there are about 4,831 compounds that are similar to benzoic acid. So, and if you look at them very well, they have that same benzoic acid structure. But the difference is that some substituents are added to it. Some other chains are added to the benzene structure. So that is similarity search. So this similarity search gives you the privilege to search for compounds that are derivatives of benzoic acid. And it operates based on the Tanimoto principle for fingerprinting. So if you want to derive if your interest is okay, let me see if since my benzoic acid is giving me a better inhibition or is having good inhibition, good activation or modulating effect, let me see the derivatives. So if you are doing this, you are going out of natural products because that derivatives you're looking for is no longer found in the plant. So this one now you are going for synthetic compounds. So at the end you have modified what you got. So how can you retrieve these compounds? There are 4,831 compounds. What you just need to do is a very simple thing. Come to download. This download here. Once you click this download, then you will see different options. It is now on this note that you can retrieve your SDF once. So I can retrieve these 4,831 compounds. I can get their structure once. I was looking for that in the last two we did, but it was there before, but I don't know why it wasn't available again. So you can actually download their structure once without you copying and pasting one after the other. What you just need to do is, what do you want to download? If you want CSV we download with their smart codes. So you have their smart codes. Then since I need their structure, very important, I will choose SDF. Then I will choose 3D. I will click on SDF, but first I will choose 3D. I want 3D conformance. Immediately you choose SDF, it will prepare those compounds to download those compounds in one file. Are we together? Hello?
Yes, sir, we are.
So immediately you choose SDF, it will download those compounds in one file. So they will be like a single file. Of course, most of us have done clustering using the OpenBabel. You can use OpenBabel to cluster those your hundreds of compounds into a single file. So these 4,831, their structures has been compressed into a single file. So you can use them and do any kind of work you want to do. So it will take time to download, so I'm not interested in that for now. All right. So this is how you can retrieve for similarity search. And when you are doing a QSAR, when you are doing, okay, I will explain how you can use PubChem also in QSAR. I will also explain how you can use PubChem in generating data for QSAR. Now, if you go for substructure, substructure here, substructure, I don't like using substructure because it will give you compounds that have the same or compounds that have similar scaffold with benzoic acid. Maybe compounds that have benzene but not benzoic acid. So that is what it gives you in substructure. So I don't like using substructure. There are more than 10,000 substructures. You can click on one for full search. It will search all the 10,000 and more substructures. Well, I will recommend you go for 3D similarity also. You can try 3D similarity. You can try 3D similarity and see what you have in 3D similarity. or most often I use just the similarity search to get analogs of benzoic acid. Use similarity search to get analogs of benzoic acid. Okay. Another thing we can do here. Another thing we can do also is, okay, superstructure now is very useless. Don't even go for superstructure. What superstructure does is a fragment of benzoic acid. It provides the fragment of benzoic acid. You can only use this if you are interested in fragment-based drug design, because we now have fragment-based. Before we used to have ligand-based and structure-based, but now we have ligand-based, structure-based, and fragment-based drug design. So what superstructure gives you? It gives you fragments of that your compound. Like in the benzoic acid now, it will break it. You have ethene, you have benzene, you have a carbonyl compound. So it will just pieces. It will pieces the benzene structure to give you fragments. Those fragments that make up the benzene structure. So that make up the benzoic acid structure. That is what the superstructure gives you. So if you want analogs, go for similarity search, and you can even sort them. You can sort them based on the property you want. Okay, you can sort based on the property you want, based on hydrogen bond count, hydrogen bond donor count, hydrogen acceptor. So you can sort based on the property you want. Okay, that is by the way. Now let me explain the tracer. Let me explain the tracer. So I just want to dwell on PubChem now. I'm giving you overview of what you can do with PubChem. Now for tracer is compounds with similar structure, we possess similar activity. So I want to do a work on, let's say, paracetamol, and paracetamol has been known to be as an anti-inflammatory drug. Now I want to see if compounds that look like paracetamol, if they can possess the same activity or similar activity with paracetamol. So what you need first, you need your training data. I'm just giving you a clue of QSAR, but we shall deal with it by February 1st week. So you need a training data. You build a model, because we want to use known activity to predict for unknown activity. So if I have 5 compounds that I have modeled myself that have similar structure with paracetamol, and I want to see if this my 5 structure can possess similar activity with paracetamol, what will I do? I will decide to use this my 5 structure as my test data. They are the ones I want to test. I want to see their ability, and I don't want to stress myself doing a in vitro study, lab work on these compounds against inflammation. I want to use computer prediction to see if they can have that ability without me doing docking. This is purely what to do in ligand-based drug design. I will look for already tested compounds that is similar to paracetamol. Already tested compounds, as much as possible, up to 1,000, 100, 200. I'll look for them. They will form my training data, because I will use them to train to build a model. In machine learning, I usually use ANN, the artificial neural network model. So we build the model, but how can I generate this compounds that have been tested already? This compound that looks like that are similar to paracetamol, that has been tested already, that has known activity. How will I generate those compounds? Just come to your pumpkin and type paracetamol and search. Once you search, this just as if you are doing a bioassay, because you do that through bioassay. You come to bioassay, and click on bioassay. So when you click on bioassay, you can sort them also. You can do the sorting based on active substance count. So you can do the sorting based on active substance count. So when you can sort, let me see if this will be fast. Active substance count, decreasing. Okay, so this was very fast. Can you see my screen?
Yes, sir.
So now what
Yes,
this
sir.
means,
Yes, sir.
Sorry.
so there are
Hello.
460. Yes.
When I type paracetamol, so what next after typing paracetamol?
You search, you hit your enter button.
Okay, click
Your
on
enter key.
search. Okay, I saw acetaminophen, paracetamol.
Come straight to bioassay. Go to bioassay.
Okay.
Yeah, and do what we did before, like sort based on active substance count and sort by decreasing. So now the first one here is showing tested compounds 41, but active substance is 37. So we are interested in the active substance, not the tested compound, because not all the tested substance has activity. You will see it. Now, since I need these compounds that are similar with paracetamol that have been tested, so I will just click on this, click on the bioassay on the report, then scroll down. Good. So when you scroll down, you will notice that these compounds are similar, and you are seeing their activity. So these compounds have similar structures. They are all having similar structures. They are all having similar structures, and they have activity. So one thing about this place is that you can see their IC50 because they are inhibiting. They are inhibitors. You can see their IC50 values. I will take time. The essence why I'm coming to this is just to show you how you can retrieve the data for QSAR, because we are dealing on chemoinformatics. But by the time we get to the tracer, and then we explain more details. So you can now download by coming to your download. You see options now. You see all active. This is active less than or equal to 1 micromolar, which is their biological activity, the IC50 values. The ones that are less than or equal to 1 micromolar. Just pick your active. So just take one that are active. So click on save under active, and the 41, the number of compounds that are active will be downloaded. So you can open them. Then you see their IC, you see their smart code and their IC50. So, but that is, by the way, when we get to February, I will then take my time to explain this. This is just how to retrieve the data. How to retrieve the data. But this is not enough to build a model. It's not enough to build a model. Yeah, it's not enough to build a model. So now you can also retrieve others. We just used one out of them. You can go back to the second report. You can go back to the second report. You come to the second report. So most times you notice that some of the compounds in the report are similar. From my experience in training students, some of them are same compound. You will now filter them. You filter out the ones, you remove duplicates. But that will be by February. So this is how you can derive, how you can download your analogs. This is how you can download analog derivatives of a particular compound of interest. We are already 4 minutes out of our time. I think we'll continue by Friday. Okay. Do you have a question? If you allow me, I will keep talking.
Yeah, we're still unable to retrieve the 3D for our main compounds or drugs. We only show us for the analogs.
Yes. So it is not there now. It is not there. So retrieving their 3D means you'll be copying them one by one and download. I'm sorry. That is how those that built PopTeam now made it. So, but that is why you can write a script. No, a friend. You can convert your. I've tried that, but it's not working. In OpenBabel, you can convert. You can convert your SDF to smart code, but I've been trying converting smart code back to structure. It's not working. But you can use scripts, Python scripts, to do that, to convert your smart code to structure. You can use Python script for that. So since you have thousands of compounds, you just downloaded, and what you got was just their smart code, and you don't want to stress yourself in going one by one. If you are good at programming, you can write a program in Python script using Python. It can convert your smart codes to structure. We don't have plan of running a training on that for now, but maybe we'll look into that later. But for now, we don't have a plan of running a program on programming. But that is where we are driving into. Science is improving day by day. If I tell people now to go and learn programming, you say it is for the computer science students. But if you want to incline in drug design, you really need programming, because now we program, since what you want to do are not found in some of these software. So you write a program that will work for you, that will do what you want to do, because there are a lot of things you want to do that are not found in some of these database and software that we have. You can now build a program for yourself. So it is good. I've already encouraged my people here to start learning, go and do programming, go and learn programming. So the same thing, I can encourage you to go and learn programming, so that by the time we bring up a training on drug design in programming, so you can be able to flow with us. All right, let's take the questions. Just 5 minutes.
Yes, sir.
All right.
Yeah. Hello, sir, doctor. Good evening.
Yeah, good evening.
Thank you, sir. I'm so grateful. After we've seen the results from Excel,
Yeah.
based on the IP50 value, I can say 34 nanomolar. That should be the unit, right? So
Yes.
34, 34, 66, 15. The one with lowest IC50 value
50
will be
is
the
more
more
effective.
potent ones, right?
Uh,
Oh,
Good.
all
Yes.
right. Thank you.
Yeah. The lower the IC50, the better. In fact, do we have people from microbiology here? Microbiology.
Yes.
Uh-huh. Let me correct. I told my friends.
Yes.
Fine. I told my friends. I told my friends
Yes.
last year this on this. Do you know that most of us who do microbial work, antimicrobial, antibacterial, antifungal, all your anti stop at sensitivity, MIC, MBC. You don't finish if you can. But I'm not wondering why can't we do IC50? Most of these people in microbiology, they don't take their word to IC50. How can you tell the effective inhibition? You just rely on your zone of inhibition. Oh, there is clearance. There is no clearance. So please, those of our friends now in microbiology, anytime you do your inhibition work, make sure you keep your data in IC50. It's very simple. Just it's a graphical representation. You can extrapolate the value from graph. That will tell you the extent of the inhibition of your different extracts, the different concentration you use in MIC or whatever, to tell you the different concentrations and their inhibition. And that is these values we got here now. We are experimental data from lab work. So if you don't take your anti studies to IC50, we cannot continue from there because we cannot tell the effectiveness of the inhibition, the extent of the inhibition. Then if your work is activating, that means we now use EC50. So even those in biochemistry, those in biochemistry that does in vivo studies, in vitro studies, we are pleading, keep your values, keep your results in IC50, EC50. Then those in even in biochemistry that is doing acute toxicity, right? Yeah, acute toxicity, chronic toxicity, and others. Take your value to LD50, latter dose. That is, it is on this point that we can be able to interpret the toxic effect of your extracts. So for those who are already doing it, thank you so much.
Yeah, I used to do it. I work on in vitro and in vivo antioxidants, inflammatory, and antioxidogenic. And after the result, I use GraphPad and Excel to calculate my IC50 graphically, and I got the results compared with the standard.
Very good, very good. That will make your work. Anybody that picks it will say, okay, that is the only way we can compare that you have done a work. Except that I will throw it away from you. All right. We have explored. Do you have questions? Let's just take the comments and interactions as we run it up for today. Okay.
Yeah, yes, doctor.
Yeah.
When we started the training tonight, I had a question based on the biodiversity of the plants, based on the phytoconstituents. And there
Yeah.
is one other thing. The period of harvest also
Yeah.
determined. So
Yes.
I had a training in OAU a few weeks ago in their Department of Pharmaceutical Chemistry, Faculty of Pharmacy. Uh, and one of the presenter said something that there was a plant in OAU during the month of, I think it was not around March or April. They harvested the leaf and determined the phytochemical analysis, and there was a particular compound that was present then. Then 4 or 5 months after, they harvested the
Mm,
plant again and discovered that that particular compound was not there.
mm,
So they
mm.
have to wait till another season the following year. When they harvested that plant again, they saw the compound there. So that means the season also determine the phytoconstituent of the plant.
Yeah, good, good, good. Thank you. Yes, yes, yes. I was at UNESCO last year. Was it October at UNN? Yes, the UNESCO had the conference there. This issue was raised and was discussed. Some guys from South Africa came.
Hmm,
So yeah, these are challenge we have, and
hmm,
that is why I said that if you want to go into business or developing a product, then you must you must be using your own. You must
hmm.
rely on your own. You
For
don't.
reproducibility, for
Yes,
reproducibility.
yeah, for the sake of possibility, you
Hmm,
must
hmm,
rely on your own. It will be hard though, but
hmm.
well, I will not share my secrets. Yeah, I will keep it. Uh, but yeah, now it's my SOP. So you you can ask God to give you your own wisdom.
Secondly, sir, the plant that I'm working on, uh, Stonebreaker. So most of the journals that I
Stonebreaker,
downloaded,
Philanthus Nehru.
Neruri, yes.
Yeah.
So most of the journal that I downloaded, so they were talking about the leaf. Can I concentrate on the leaf instead of
Yes.
the other parts? All right,
Since
sir.
that is what you found. Since that is what you
Oh,
found. Yeah.
yes.
Since that is what you found. Yes.
I've run some analysis on it, HPLC, GCMS. So
Or
I can't include by my own my own results too. Since I'm not working on the experimental constituents. We're working on
yes.
review literatures.
You can just leave your own.
Oh, all right, sir. Thank you, sir.
Okay. Both.
Hello.
Yeah. Yes, sir.
Okay, I just wanted to make a comment on the last speaker, especially seasonal influence on the phytochemicals in the plant. Yes, that is it affects phytochemicals of a plant. But again, apart from that, even if you have maybe the presence of some kind of ants on that plant, can actually modify the phytochemical present. The plants, the ants that gather around the plant or are, that prey on that plant can also affect the phytochemicals that are expressed by the plant. And that is why most journals now they will ask you to give the GPS coordinates of the place of collection of the plant because they want to know because the place of collection can play a large role on the phytoconstituents of the plant. If there are animals around that area or that live in that plant, they can also affect the phytoconstituents of that plant. So that these are some of the considerations that you take note of, and then of course you use them in your reports. I just wanted to say that. Thank you, sir.
Thank you very much, sir.
So you see that there are a lot of conditions. Yeah, there are a lot of conditions that we must keep. But those conditions can be managed. They can actually be managed. Yeah. If we really want to do something, they can be managed. So they should not discourage us. All right. Thank you very much for coming around tonight. Tomorrow we are starting molecular modeling. The training is Wednesday and Thursday. Since this week we had the training yesterday. So tomorrow I will encourage us to register in that molecular modeling. One thing that we'll do is it's just similar to what we did in the Macro 4, but we are extending. Is that we are going to teach you how you can do high throughput screening. That is not docking yet, where you can identify your lead before docking. The reason why I brought that idea up was I handled a training in Genomark late last year. So one of the participants raised a question that is it possible that after we have gathered 500 bioactive compounds, because I always shout for 1,500, after I have gathered 1,000 bioactive compounds from my plants, will I dock these 1,000 bioactive compounds? That was what she asked. And a professor from this Ebenidion University, Okada, I think so. So, will I dock the 1,500? Can that be a way of reducing these bioactive compounds before docking? When she asked that question, I was like, I have not really looked at that, but I told her that in our next session we'll provide an answer. So the answer is, if you are an expert, you don't need to dock this 1,500. You just have to look for the ones that are drug-like, using their properties, filter them off. At the end, out of this 1,500, only 10 or 50 might even pass the test. Then you work with those 50. The rest may not possess drug-like properties. So you do a thorough screening, and that is what we shall be exposing us to Wednesday and Thursday and more. Modeling molecules, designing them to possess drug-like properties. What we did in Macro 4 was to design them, but now we are doing screening, because in the Macro 4 there are two aspects: designing and screening. So if you can join us Wednesday and Thursday as we screen bioactive compounds, so we can identify our lead for docking. Once more, thank you for your time.
Sorry, so for some of us that will wish to join tomorrow's session, we don't have the email and the link and the link yet. So if you can please help us to send it.
Okay, I will send it. We always update it in our announcement platform, but I will send it to this platform.
I think there are some materials that we have to have as well in terms of software. Am I right for tomorrow's session?
Yes. So if I send it tonight, if sleep will not overcome you, you register, then by tomorrow morning very early, you receive your mail. From the mail, you can download the software before 8 p.m.
Yeah, I registered a couple of days, but I've not seen the mail up to now. Maybe you should to review, because I won't be able to register again once I've registered once.
Okay, we ask that those who have not received their mail should drop their email.
I've done that.
Today?
Yes, later today.
I'll do it again.
I'll do it again.
If I have done that later today, okay, that means we will attend to that. Or you can still.
Yeah. Sir, please, those who registered this evening, I registered today.
You receive your mail this night.
Okay.
I will try and conquer sleep. Okay. The fact is that a hand there, we don't sleep.
I beg, try and sleep.
We'll try until we achieve our goal. Once more, we say thank you. Thank you all. Thank you all. Good night.
Good night.
Good night. Thank you.
Yeah. Yeah. Good night. Thank you. Thank you.